to the next presenter, who is gonna present a project that lies well deep in my heart. I have been working on antivirals since I can remember, basically. It's a wonderful project. It's something that I find very, very exciting and very, very interesting. You see, it's now 15 years since my PhD that I started to work on the design of inhibitors for single-stranded RNA viruses. I knew nothing about coronavirus back then, but it turns out that HCV, dengue, Zika, yellow fever, they share the same genomic organization with coronavirus. But it really makes me feel great and proud that, yeah, exactly. And that's Eric adds, these are zoonotic viruses. They can be passed on from animals to humans. As we now all know, they pose a threat to the society. So I was saying that it makes me feel wonderful. It makes me feel it's a big accomplishment for my scientific, I don't know, life that we have the amazing EO, a new recruit in the lab, a wonderful student she's taken over. And hopefully we're gonna learn wonderful things soon through her motivation and research. So EO today is gonna present computer-aided drug design and pharmacophore modeling pipeline towards the discovery of novel anti-ebola agents. So please, EO, stage is yours.
Thank you. I'm sharing my screen. I think you can see it.
Yes, thank you.
Okay, so I know that Ebola virus has lost its crown as the hot topic of the day because of SARS-CoV-2, but it remains a very important pathogen concerning public health. So to give you an overview, the genus Ebola virus is part of the Filoviridae viral family, and it comprises of 6 species, out of which only 4 have been associated with human disease. And out of these 4, the species of Zaire Ebola virus, also known as Ebola virus, is the one that is the most virulent and the most associated with the Ebola virus disease. Now, for the routes of transmission, we presume, based on scientific evidence, that the reservoir host of the Ebola virus is bats. And the transmission from bats to humans and animals is carried out through a spillover event, which is the event of an infection of an animal or a human through the contact with the reservoir host, which in this case is the bats. And of course, this happens either through the preparation of meat of these infected animals for eating, or through direct contact with the bats themselves. And of course, we have the transmission of the virus between humans, and this can happen through infected blood or other bodily fluids such as semen or the placenta or the mother's milk. It can also happen through contact with contaminated surfaces by these aforementioned bodily fluids. Transmission can also happen through the handling of dead people who have died from the virus or the handling of contaminated medical instruments. And the virus itself enters the host organism through breaks in the skin or through mucosal membranes such as the mouth, the nose, or the eyes. After the infection, the virus targets primarily liver cells, endothelial cells, or immune cells such as macrophages, And the attack on endothelial cells and the infection and the consequent breakdown of the endothelial cells which line the interior of vessels, of blood vessels, is the thing that ultimately causes the hemorrhagic fever that is the Ebola virus disease. And this disease, which is a very serious and often lethal disease, has an average of 50% case fatality. Now, since the virus's discovery in 1976, there have been multiple outbreaks, mainly in the African continent, but the largest outbreak to date and the most fatal was the 2014 to 2016 West Africa Ebola virus epidemic. And it was caused by the Zaire Ebola virus species, and it had more than 28,000 cases, out of which more than 11,000 were deaths. And of course, there is an ongoing outbreak in the Democratic Republic of the Congo, and namely in the Equateur province. So when faced with the Ebola virus disease, we have a trifecta of three paths. We have the supportive treatment, the experimental therapies, and the prevention route of the vaccine. The supportive treatment, which is the first course of action, is aimed mainly at the management of the fluid loss and the electrolyte loss that is caused by the onset of the disease, as well as the treatment of various symptoms, such as the fever or the gastrointestinal symptoms or the aches. And now, in order to talk a bit about the other two parts of the trifecta, we will have to, I will have to give you a brief overview of the Ebola virus genome structure. So as we know, the Ebola virus has a single-stranded, linear, non-segmented RNA, negative sense RNA molecule as its genome. And the RNA, the viral RNA, has these genes, and these genes code for a variety of proteins, such as the nucleoprotein, which encapsulates the viral genome, the polymerase cofactor, the matrix protein, the very important glycoprotein, which is the only surface protein that protrudes from the virion of the Ebola virus. We have the factor that influences the activation of transcription. We have the nucleocapsid associated protein, and we have, of course, the catalytic subunit of the polymerase complex, which mediates the replication of the viral genome. Now I'm going to talk a bit about the viral life cycle, just in brief. We have the attachment of the virion to the cell surface, and this is done through the attachment of the glycoprotein on the surface of the virion with various host factors that can be, for example, lectins. And after the attachment, we have the endosomal uptake of the virion. And in order for the nucleocapsid to be released into the cytosol, there has to be a fusion between the viral membrane and the cellular membrane of the endosome. And that is done again through the glycoprotein, which is cleaved after the attachment into its fusion-ready form. And after cleavage, it interacts with host transporters, such as NPC1. And this interaction leads to the fusion of the membranes, the release of the nucleocapsid, and then we have the stages of the RNA, the viral RNA replication. We have the primary transcription, the secondary transcription, the assembly of the virus proteins and the genome into the new virions that ultimately bud from the cell. So now, in terms of a preventive route, we have a milestone in the history of Ebola virus, which is the approval of a vaccine. And the vaccine, commercially known as Ervebo, but known in the scientific community as RVSV-ZIBOV, is an attenuated vaccine, which is partly the backbone of a vesicular stomatitis virus, where the glycoprotein has been substituted with the glycoprotein of the Zaire Ebola virus. And while this is being used, it of course has its limitations, which are the fact that it only offers protection against the species of Zaire Ebola virus and not other species, and the fact that it's used only on adults. And we have a second vaccine that is a double vaccine with a prime dose and a booster dose afterwards, which was approved later than the first one in the EU in July of 2020. So now to a very important part, the experimental therapies. The basis of the experimental therapies when it comes to viruses in general is usually guided by the steps of the viral cycle. And usually key steps are the ones that call for the design of the inhibitor or anything that we want to use as an antiviral strategy. So we can have inhibitors of attachment. As explained earlier, we can have inhibitors at the stage of the viral fusion, of the membrane fusion, and in essence, not allow the nucleocapsid to be released. We can have inhibitors in the stage of the replication, in the stage of the assembly, or the budding. And of course, the appropriate proteins of the virus can be the targets of the antiviral strategy. An example of treatments that have been used and are being used is monoclonal antibodies, which target the entry stage of the viral life cycle, and nucleoside analogs that, for example, inhibit RNA synthesis. Remdesivir was being used, but is now has been shown to be less effective than the treatment with monoclonal antibodies. So, for example, it is being looked into as a repurposed drug for COVID-19. And so on, we can hypothetically target various stages of the life cycle with an appropriate molecule. Just to better elucidate my point, I can give you the example of the MAB114 monoclonal antibody treatment, which takes advantage of the stage of membrane fusion in the viral life cycle, because you can see here when we have the attachment of the virion to the cell surface and the micropinoketosis, we have to have the membrane fusion in order for the nucleocapsid to be released into the ketosol. So this antibody binds to the region of the glycoprotein, of the viral glycoprotein that would normally interact with the host cell NPC1 receptor, which is a cholesterol transporter. So in case of having the normal interaction and the membrane fusion, the interaction is avoided by the binding of the antibody on the GP, to the GP. And so we have inhibition of this specific stage in the viral life cycle. So in the race of developing antiviral drugs against various viruses and as well as towards Ebola, against Ebola, we have noticed a spike in the studies that are being published, especially after 2015, which are aimed towards an in silico or computer-aided approach. And these strategies include pretty standard in silico strategies, such as molecular dynamics, flexible docking, combined efforts of the two, pharmacophore design, and pretty much any standard in silico drug design procedure. Another key point in the use of in silico systems towards the discovery of antivirals is the fact that we have vast libraries of drugs, of compounds that can be either natural or that can be already approved drugs. And it would be a monumental mistake not to take advantage of these libraries towards the design of novel drugs or inhibitors. So a very important step and a very productive step is the virtual screening of these libraries in search of inhibitors that could be used in the case of Ebola virus. And to illustrate this point, I give you an example of two studies that are about 2 years apart, 2015, 2013. And one, the top one, describes a very standard approach for the screening of FDA-approved drugs in order to find new inhibitors. And the bottom study presents an in silico approach for the virtual screening of experimental drugs. So we have this difference here. You can see the standard approach with the in vitro and in vivo studies in mice could use 1,000 FDA-approved drugs in the process of the screening. And in the process of the screening using in silico tools, we have a 6-fold increase in the number of the drugs that can be used. And these were from the database of Drug Bank. And the more recent study from 2019 shows the exponential increase in the number of compounds that can be handled through in silico methods. Here we have more than 1 million compounds that can be screened in search of inhibitors that in this case target the glycoprotein. So we can see how the advancement of the computational methods has an effect on the range of data sets that can be used. Another key point in the use of in silico methods when it comes to Ebola virus is the fact that it's been categorized as a category A bioterrorism agent, which means that there are very strict and very high safety grade levels in order for Ebola samples to be investigated in a scientific way. So in silico methods offer us a way to bypass this difficulty. And of course, in silico methods give us the benefit of taking advantage of big data, which is a very important component of bioinformatics as we know it now. And as I mentioned before, big data in the form of already existing compounds or natural compounds that can be screened for the discovery of novel inhibitors. This can be done only through in silico approaches. The range and the grade of the data size itself is very is unforgiving when it comes to traditional approaches. So we have the bottom line that, as I'm trying to illustrate in the next GIF, computer-aided drug design against Ebola is summarized like this. We have the standard design and we have computer-aided. So the end goal is the same. We want to jump through the fence, but in one case, the approach is faster and safer, and in the other case, we have all these problems. So in conclusion, I would like to thank everyone in the Dark DNA group. I would like to thank my supervisor for trusting me with a general topic that is very close to his heart, and I hope I will not disappoint him. And I would also like to thank the committee of the EMBnet for having me here because it's a great opportunity. Thank you very much.
Thank you, Io. Very nice presentation. So, any questions? I can see that Domenica has written something.
Yes, that is a very good question, and it was something that I was thinking about this morning. What we can gain from the Ebola virus epidemics and study that can be used for COVID-19. I think one of the key points, and it's not exclusively related to the scientific part is the public perception of the threat and their willingness or not to adhere to guidelines. Because in the case of Ebola, we had people who, due to socioeconomical reasons, were reluctant to follow the guidelines of their medical officials, and this led to an increase in transmission of the virus. And in the case of COVID, we can see that the denial of the use of masks or the lack of willingness to adhere to the guidelines of the state and of the medical community leads to increased transmission rates, which could be avoided. So that's an important parallel between Ebola and COVID. Yes, and to answer what Eric is saying, there have been some papers that underline the fact that countries affected by the Ebola epidemics were better prepared to handle COVID cases.
Perfect. Thank you very much. Couldn't agree more.